All right, everybody. Unfortunately for you, it's not Mrs. O'Donohoe this time. It is me. I have ran out of shares on Ed Puzzle, so I can't use Miss O'Donohoe's anymore, so I got to make my own. Kind of stinks for me, and it probably stinks for you too, because she's done this a lot longer than me. But, anyways, we're going to continue on through the digestive system. Right now, we're going to talk about the pancreas. It is right there in our little diagram. All right, so I'll be honest with you guys. I'm going to tell you what I think you're going to need to know, basically because I've seen the test. I don't have it memorized, but from what I can remember, I'm going to tell you what you're going to need to know, and I'm going to kind of glaze over what you don't need to know. First of all, the pancreas is considered both endocrine and exocrine in nature. When you talk about endocrine, it basically means that it's releasing its products into the blood. So sometimes the pancreas releases its products into the blood. We'll talk about that in a second. And when it's exocrine, usually you think that means it's going to release it outside of the body. In this case, when it's talking about exocrine, it's actually releasing it into the GI tract, the gastrointestinal tract, which basically means it's going to release its products right there into the start of the small intestine called the duodenum. So in its functions as an exocrine gland, the pancreas, if you're looking at the top right, it's going to release through this duct right here. It's a common bile duct because the liver secretes the bile through basically the same duct. They come through the same little opening right there into the duodenum. Now, I know it says right here that it opens 4 inches below the pyloric sphincter. Remember, the pyloric sphincter is the little valve between the stomach and the small intestine or the duodenum. Obviously, you don't need to know 4 inches below. I'm not going to ask you a multiple choice question of how many inches below, A2, B3, C4, D5. I'm not going to ask you that. So you can write it, and it's good to know that it's about 4 inches, but certainly do not try to memorize that. All right, so the histology of the pancreas. If you didn't know or can't remember, histology means the study of tissues. So this is basically, hey, what tissues are in the pancreas? And we're going to start with the islets of Langerhans, named after the German scientist. I forget his first name. I think it's Paul, who discovered these back in the 1800s. And this is what's going to be the lighter colored tissue that's in the pancreas when it's stained. Will you need to know that? No, but this is where it's at. So these are the islets of Langerhans, also called the pancreatic islets. This will become important. This is the portion of the pancreas which is endocrine in nature. That means it's going to release its products into the blood. There are basically 2 types of cells in this lighter region right here. Alpha cells. These produce a hormone called glucagon. I'm not going to go over it too much because the next slide is going to talk about it more. This is what increases blood glucose levels when they are too low. And then you also have beta cells, which produce insulin, which decrease the level of glucose in your blood if they get too high. Now, people with diabetes have trouble regulating this process of keeping your blood sugar, getting it higher when it's too low or bringing it down when it's too high. But definitely remember that glucagon is going to make your blood glucose level increase, go up, and insulin is going to make the level of glucose in your blood go down. All right, the remaining part of the pancreas, the vast majority of it, about 98 to 99%, is made up of what's called acini, and that is the darker portion of the pancreas when it is stained. And that is the part of the pancreas which is exocrine in nature. So it once again, it's not releasing its product outside the body, but it is going to release its product into the GI tract. It's going to release it into the small intestine, the duodenum, and these are the products, these enzymes, which are going to help in digestion. All right, so this is the exocrine functions of the pancreas. So this is what the pancreas is going to release into the duodenum of the small intestines, and it's going to release what's called pancreatic juice. Now, sure, it's clear, but you're not going to be asked about that. It contains water, obviously. Almost everything contains water. The things you need to know, it contains enzymes, and it also contains sodium bicarbonate. Sodium bicarbonate is basically baking soda that you can buy at the store. And sodium bicarbonate is a base. When the chyme, the food from your stomach, reaches your small intestine, the duodenum, it is very acidic from the hydrochloric acid that your stomach releases to digest mostly proteins. Well, if you're ever going to neutralize an acid, you add it to a base. Because when you add a base and an acid, you're going to get water and you also get salt. So basically, the base will neutralize the acid. And that's extremely important because the stomach is built to survive in an acidic environment. It likes the acid. Your small intestines, on the other hand, if it wasn't neutralized by the sodium bicarbonate, it would quickly burn a hole right through your small intestines. And obviously that would not be good. So these enzymes that the pancreas release into the small intestines are going to be pretty important. These are them right here. First of all, pancreatic amylase. Remember, we had salivary amylase that is released in the mouth, and it digests starch into sugar. Well, pancreatic amylase does the same thing, so this breaks down starch. You get pancreatic lipase. Some people call it lipase. I don't know why, because it digests lipids, and that's the pancreatic lipase digests the fats. And you have 3 different types of enzymes that digest proteins. Anytime you see any kind of trypsin, whether it's just plain trypsin or chymotrypsin, always digest proteins. And it also releases carboxypeptidase, which also digests enzymes. Now, these are also released in their inactive form, kind of like pepsinogen in the stomach, and they're activated in the intestine by an enzyme called enterokinase. Are we going to ask you about that? I don't think so, but just in case, there it is. But once again, don't go crazy trying to memorize that. And also, just for, just to be thorough, they also release these enzymes right here, which helps you break down nucleic acids, which is DNA and RNA. Those are your nucleic acids. So when you eat stuff like meat, obviously you're going to eat, in plants and anything you eat is going to have DNA or RNA in it. And you got to break that stuff down too, and those 2 enzymes are what does that. Obviously, the DNA and RNA in food is very, very small, so these are not going to be that important. Concentrate on the enzymes that break down fat, proteins, and carbohydrates. All right, so these next couple of slides are going to be really, really important because on our exam, I think, which is going to be next week, end of next week if I'm not mistaken, this negative feedback loop of blood sugar levels is going to become you guys' favorite type of question. Is that multiple choice? Nope. Is that fill in the blank? Nope. Is that an essay question? Ding, 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 ding. Yep, it's going to become an essay question. So you might want to take a little extra notes on this and maybe study this a little bit harder than most things. All right, so the negative feedback loop. Remember, negative feedback. Negative means when the stimulus, whatever stimulus the body receives, the body's going to do the opposite. So if the blood sugar levels get too high, your body's going to bring it down. If the blood sugar levels get too low, your body's going to do the opposite and raise it back up. Well, all right, so we're going to start out with low blood glucose levels, which is also called hypoglycemia. It's detected by your pancreas, and it causes your pancreas to secrete what's called glucagon, and that is done by the alpha cells in your pancreas. Now, glucagon is a hormone, so it's going to go into your blood. So this is the endocrine function of your pancreas. So your alpha cells release glucagon. Basically, it acts on your liver, mainly your liver, because your liver is what stores the vast majority of what's called glycogen. Glycogen is how animals store their energy. Plants store it in starch or in oils. Animals store it in glycogen, and we also store it in fat. So when the pancreas releases glucagon, it causes the liver to break down the glycogen it stores in its tissues into glucose, which is released into the blood, which causes the level of glucose, sugar, in your blood to go up. So in a nutshell, when your body detects that the blood glucose level is getting too low, which is hypoglycemia, your pancreas release glucagon, a hormone, which basically tells your liver, hey, we need more glucose in the blood. So please start breaking down all the glycogen you have in storage, break it down into the smaller glucose and release that into the bloodstream, please, because we need more glucose in our blood. Now, if your blood glucose gets too high, too much sugar, too much glucose in your blood, which is called hyperglycemia. If you have too much energy, you say you're hyper. Well, if you have too much sugar in your blood, you're hyperglycemic. It's hyperglycemia. Well, this is going to inhibit the release of glucagon from the pancreas, and it stimulates the release of another hormone called insulin. This insulin is secreted by the beta cells in your pancreas, and that tells your cells, please diffuse or take in glucose, especially cells like skeletal muscles, because skeletal muscles, they need glucose because your muscles need a lot of energy. So it's basically the insulin is telling your skeletal muscles, take in the glucose and please use it because we have too much of it in our blood. It also tells your liver, hey, all that glucose in the blood, let's start storing it again as glycogen. It also tells your liver and your other cells in your body, let's start making proteins. The reason for that is the process of making proteins takes energy. So whenever you need energy, you need glucose. And when you're using glucose, the level in your blood is going down. And all of this activity will cause the blood glucose levels to fall. If it falls too far, goes too low, of course, then we'll go back up here to the alpha cells, which will once again release glucagon to increase the amount of glucose in the blood. All right, so here's basically what the last slide said in kind of picture form. So start over here at 1, if blood glucose levels start to get too high, hyperglycemic, your pancreas, the beta cells, release insulin. That tells your liver, hey, start taking that glucose and please start storing it again as glycogen so we can get rid of some of this glucose from the blood. It also tells your cells, mainly your muscle cells, please take in some glucose and start burning it so we can get some of it out of our blood so we can return back to homeostasis. And the opposite of that, if glucose levels get too far down, too low, called hypoglycemia, That tells your pancreas, the alpha cells, release, please start releasing some glucagon. That tells your liver, hey, you got all that glycogen, that stored energy inside of your tissue. Start breaking that down, turn it into glucose, which then enters the blood and will return back into homeostasis by raising the level of glucose. All right, so the histology of the liver. Once again, you guys know what histology means, right? It is the study of tissue. So basically, it's saying what kind of tissue is the liver made out of? Well, you're going to need to know this. Hepatocytes, those are liver cells. Might want to put that in your notes. Will it be an essay question? Nope. Will it be a multiple choice question? Probably. I'm going to breeze through these two because I don't think they're on our test, but who knows. The bile canaliculi, those are just the small ducts between the liver cells, the hepatocytes, which collect the bile and empty them into the small intestine, the duodenum. The hepatic sinusoids are just blood capillaries between the liver cells, the hepatocytes. Now these are important. The Kupffer cells. Once again, that's the guy who discovered them. These are the type of cells in your liver which basically detoxifies your blood. So these are the ones that worn out blood cells, they get rid of them. Microbes, get rid of them. Alcohol, toxins, it detoxifies them and basically keeps you healthy. This is why if you take too many drugs, alcohol, even over-the-counter drugs, it can destroy your liver because it actually is hard on the liver to detoxify toxins. And these cells right there are what does that. All right, so bile. The liquid, which yellow-green liquid, which is secreted by the liver, stored in the gallbladder. That is why every time you see on our little diagrams, the gallbladder is always green. That's because it stores bile. It is important in the emulsification of fat molecules. Fat globules can be very, very large. Before they can be digested, broken down even further to be used as energy, they first need to be broken down into smaller pieces called triglycerides. Once they're into triglycerides, then they can be broken down and absorbed by the small intestine and used as energy. Now, this other term, bilirubin, that is just a pigment, and pigment basically just means color. So bilirubin it gets its color. It says heme here, and that's true, but heme is a part of what's called hemoglobin. And that is a part of the red blood cells, which carry oxygen, and this is the part of your red blood cells that carry oxygen. So the breakdown of hemoglobin gives you heme, which then gives you the color of bile. It also gives you the color of what you excrete out as waste. So that's what makes your feces look brown, and it's what makes your urine look yellow. It just depends on where the hemoglobin is being broken down. All right, so some functions of the liver. Metabolize carbohydrates. I don't know why that was yellow. Let's turn it back to black. What you need to know here, what we said earlier, is this right here turns excess glucose in the blood and turns it into glycogen, which is where energy is stored in the liver. So if glucose levels get too high, the pancreas inform the liver, hey, start storing that again as glycogen. It also helps to metabolize lipids, not by digestion, but through emulsification, which breaks down the large fat globules into triglycerides. It makes them smaller, which then they can be broken down, digested within the small intestine, and then be used as energy. And more functions of the liver. It helps metabolize or break down proteins in a process called deamination. Don't worry about it too much because I don't think it's on our test. Deamination means it just removes what's called an amine group or an NH2 from the amino acids, so then it can be used as energy. Liver is also very, very important. Your body produces ammonia every day, which is toxic, and it converts it into urea, which then can be excreted as urine. And it also helps to synthesize certain proteins like platelets, which are used for clotting your blood if you're bleeding, and also for some immune system functions. Once again, don't worry about that too much because I don't think it's on the test. But there it is. All right, our last slide. Yay. All right, so some more liver functions. A huge function of the liver, detoxification or detoxifying the blood. So everything, not necessarily bad. I mean, drugs, you think of drugs as bad, but sometimes you need painkillers or whatever, over-the-counter drugs, alcohol. Your liver is what detoxifies, basically makes them harmless or gets rid of them. Even hormones. So too much hormones in your blood, even if you take birth control pills, it's what your liver is what takes care of that and gets rid of excess. We went through bilirubin. It stores vitamins and minerals, mainly your fat-soluble vitamins, which is, I say ADEK, A, D, E, and K, but also Mr. B12. It screws up my ADEK, but ADEK plus B12. Those are fat-soluble vitamins. There are also water-soluble vitamins like vitamin C. These fat-soluble vitamins, you don't want to take too much of them because it can be bad for you. Water-soluble, like vitamin C, basically you can take as much as you want. Like I take Emergency, has like 10,000 times is what you need. But it doesn't really hurt you because it just goes out your urine. But if you have too much fat-soluble vitamins, it can be harmful. And it also helps to activate vitamin C, which is what they call the sun vitamin. And those are the functions of your liver.